Good morning, everybody. And whilst we're all sharing, I think I got into marine biology because I watched the movie Jaws when I was 7 years old. And I discovered that you could have a job doing this marine biology stuff. And then years later, someone said to me, if you're a marine biologist, you'll never do a proper day's work in your life. That's totally untrue, as we all know. But we all have a good time. Anyway, I'd like to talk about coral reef reproductive success or failure. And this is work that's been led, it's a collaboration between UQ and CSIRO primarily, and it's been led by the postdoc Jared Ricardo, who does a fabulous job. And we're all aware that if you've got a healthy reef system, you'd expect there to be vibrant reproductive success. And the concern that many of us have is that as reefs degrade, corals become farther and farther apart, and this might mean it's less likely that they achieve successful fertilization. And the reason for this is that there's a strongly nonlinear relationship between the fertilization success of eggs on the y-axis and sperm concentration on the x-axis. And so any process that dilutes sperm, such as corals being farther and farther apart, means you're much less likely to achieve reproductive success. And this is obviously a question that might influence how you would output corals. You want to make sure that they're outplanted at distances that mean they're close enough that you can achieve some successful natural reproduction. But it's a really difficult thing to study because if you do it in the field, there are so many confounding influences. You can have other corals that will hybridize with the corals that you're interested in and so forth. And so the approach that we've taken is to work in isolated lagoons where we can control the process. We can transplant colonies from out on the reef into the lagoon, we can control the spacing, and we can test for any confounding effects and hopefully reject them. And so we've been doing that both at One Tree Lagoon, which is fantastic because as the tide falls, it isolates the system, and also at some sites in Palau, Micronesia, where we get another shot at doing spawning experiments later in the year. And the sort of general setup is we create these beautiful little condos for corals, and any coral would be very happy to live there for a short period. And you have the egg bundle collector above it, and on the top of that bundle collector, we have a... Oh, no, we don't. Okay, on the top of that bundle collector, we have this little pot. It's about 15 centimeters high, and the walls are made of plankton mesh. So once we seal it and it's collected the egg bundles, the eggs are forced to stay in the pot, but the sperm can wash in and out so that those eggs can be fertilized. And you can see in that panel on the top right, this is in the daytime, just to give you an idea of what it looks like, that this thing will float on the surface. We attach light sticks so that we can track them at night. We'll talk a little bit in a moment about this strange green stuff. And then we can follow these floaters for at least an hour at night once they've spawned to see what kind of fertilization success will have occurred. We bring them back to the lab and quantify success. So the first experiment we did was at One Tree, and it was a really small-scale experiment. We just had 8 colonies of Cropera tenuis. We separated them by 2 meters, which is a pretty reasonable distance. And we asked, would you see any fertilization success? And to be honest, I was amazed if we would see anything. Bear in mind that when those bundles are released, you have to wait at least 15 to 20 minutes before those eggs are ready to be fertilized. And that's quite a long period of time for those eggs and that sperm to go in different directions, especially when you're starting with such a tiny pot of corals. What we actually found was sort of close to that. We did find success. There was about 4% on average fertilization success in these corals. So this is fertilization success on the y-axis. And to be honest, I was even surprised that we got anything happening. We then, on that same night, did it with a second species. This is a Cropera digitifera. Now we intensified the spacing from 2 meters to 1 meter. We had more colonies, and we got slightly higher fertilization success. We then did it in Palau with another species. This is a Cropera hyacinthus, 1 meter spacing again. Again, a pretty small patch of colonies, of 20 colonies, and we see a slight increase again. So what does this tell us? Well, it tells us that if you have a really tiny patch of corals that you've restored, remarkably, they do actually contribute a small amount towards the population through fertilization and the generation of larvae. But of course, you're getting pretty small fertilization success. And the real value of these experiments was to help us to parameterize a model that we could generalize. So the following year, we did a more extensive experiment. And this was done in Palau. And if you look carefully in that top left panel, we have a dense patch of colonies that are at the density that we find on the reef. This is Acropora hyacinthus again. And then we have a series of colonies on their condos at different distances, so at 0.5 meter, 3 meters, 10 meters, and 30 meters, in different directions. And again, the idea was, if we have a spawning event and you've got this central high-density patch of colonies, what's the likelihood that these colonies that are farther away are going to achieve fertilization success as we get further from the main center of mass? And I'm not going to show you the results yet. I mean, obviously, because it's not going to let me do anything yet. But anyway, yes, we'll move forward. So the reason we did this was, again, to parameterize a model. And Jared's done a great job here, taking a model that was originally created by Teo and Pete Todd in Singapore, and then extending it. So this is parameterized, in this case, for small Acropora. And we spent considerable time, along with one of the PhD student, Liz Buccheri's work, looking at fertilization dynamics and modeling the dispersal of sperm and eggs. And so when you get this model, you orientate the corals in some kind of grid on your model. You then track what happens with individual corals. They release their bundles of eggs and sperm. They hit the surface, break apart. They then start to disperse. The eggs are floating. The sperm are being able to mix in 3 dimensions through the water column. There can be advection and diffusion. And then there's the fertilization kinetics, and that will vary according to sperm concentration, egg size, and so forth. There's quite a lot of complexity there. And to try and get a handle on what the sperm dynamics were, we were releasing these harmless green dyes at the same kind of tidal conditions during the daytime, of course. And then you have a drone sitting above you, which allows you to trace how that sperm is diffusing over the seascape over the next sort of 20 minutes or so. We can then use that to create the model. And this is Jared's funky little model in R, which you can see a little simulation there. Well done, Jared. In this case, it predicted 8% success. Very, very nice. So what do we see when we do that? We see generally fertilization success on the y-axis again. The gray bar is the observed data for these 3 species and these small-scale studies. And the blue circles are some of the model predictions, depending on how you're working with that model. And you can get a variety of outcomes from that model, but in general, they show the same sorts of patterns and fairly low success under these circumstances. And you can do various sensitivity analyses that I won't describe. And then you can try to generalize, and this is the key thing. What do you learn from this? So if we look generally then, on our y-axis is the size of the colony. Obviously, bigger colonies, on average, will tend to be more successful. They release more gametes. And on this axis, we've got the spacing, the distance between colonies. And you would expect the highest fertilization success with big colonies that are close together, which is the upper left-hand corner, which is exactly what you would predict. And it tails off as you get further away. And this was our goal, to have something that we've now, based on real empirical studies, that we can start asking questions regarding how does spacing affect reproductive potential. But we also wanted to test this out, and I've just returned from another experiment in Palau. This time we did it not with an experimental manipulation in the lagoon. We wanted to know what happens actually on the reef itself. So we're going to one of our study sites here looking at Acropora hyacinthus, and we're looking at corals of different distances, different separations. And these are some pretty high-density corals. So here's the experiment in action. We've got the collectors on top of the corals. They're at different separations, and this is what we find. So again, the y-axis is fertilization success. The x-axis is the nearest neighbor from your colony to another colony that's of the same species that's spawning that night. And we did this for 2 successive nights. The first night was really calm, perfect spawning conditions. The second night was very windy, and everything changed. The success rates plummeted when we had windy conditions. And this is actually the topic of Liz Buccheri's PhD thesis. But that's what we saw out there at night. But the overall trend that you see is what you would expect. As you separate these colonies, fertilization success declines. Interestingly, the models predict that we would have something like a 20% or lower success once you separate them by around 6, 7, 8 meters, that kind of distance. And that is what we're seeing on the reef itself. So I think models will never be a perfect match to data, but it gives us some sense that at least for acroporids, that there are some of these critical spacings that might be important. So what does this tell us? First of all, I think that coral separations of 8 meters or more are going to tend to lead to low fertilization success in reproductive events naturally. Once those corals get separated by at least 12 meters, I think we might expect very low fertilization success, at least in the acroporids we've worked with so far. And I think that this can help direct some of the activities. You know, one obvious implication is if you were able to go and assess the density of colonies on a reef and find that they're at lower density than this, then perhaps one goal of restoration is just to add enough density to reconnect that population and switch the reproduction back on again, rather than allowing it to fail. But this needs to be done with more corals. And lastly, I'd like to acknowledge all of our partners and collaborators, in particular the Palau Coral Reef Centre. Thank you.
Great stuff, Pete. And I saw my name there, Adriana, in the acknowledgement, so that's good. My other name.
Well, you're everywhere, man.
Right. So I think this is very critical for our RAP in terms of the density at which we deploy or our RAP deploys. That's really useful. But it can be, in my view, a bit worrisome as well, because one of the objectives of what we are trying to do is to spread a particular genetic configuration that we are putting in a dense patch. And then if we just let them spawn and go through connectivity, we know that the proportion of them that are going to survive is small and they're going to go everywhere. And in a space, in a reef that is getting more and more degraded, the likelihood that they're going to happen to land and grow in a place where they are going to be close by other colony-specific is probably small and getting smaller over time. So I wonder if these results also suggest perhaps the potential of a rethink of how we follow up once we deploy, and whether we should be perhaps collecting that output of a dense space and put it elsewhere in a dense space as well, as opposed to be counting on connectivity.
Yeah, that's a really good point. And I think it clearly argues that, and this isn't going to surprise anybody, and it's not a new thing to say, that restoring at low densities, it's going to be probably a waste of time from a reproduction standpoint. It might restore some short-term ecological functions, but it won't switch the ecosystem back on again. How we deal with these problems of trying to maintain the sort of genetic composition of corals that are outplanting and how that gets diluted is a hugely complex question that I can't answer in a few minutes, of course. And whether that argues for trying to create high-density patches of equally thermally tolerant corals that go out there, rather than sort of spreading them out, you have them in concentrated patches so that they do at least interact with one another, would make sense if that's what you're trying to do. But one of the real problems we have here is that hardly anybody assesses the density of coral species. And so even just trying to get data on the density of individual coral species across a reef is difficult. There's very few data sets that have done that. We work on coral cover, life forms, and so there's a lot we need to know to be able to operationalize this more effectively.
I think we only have time for one more question, sorry.
Thanks, great talk. I'm just wondering, for the studies where you translocated the coral broodstock, how synchronised was the actual spawning on each night, and would you expect fertilisation rates to increase if there was 100% of the broodstock that spawned?
Yeah, good question. So interestingly, the typical thing that we see is that on one night you get a dribble of a few colonies, and the next night they all go. So we did have high synchrony in that regard. When we did it in the field just recently, again, the first night was a combination, and we recorded this, of course, a combination of a bit more than a dribble to moderate spawning of colonies on that first night during these perfect conditions. And then the second night was when everything went very strongly. And although a couple have gone completely only on the first night, most went both nights and more strongly on the second night. And yet that's when we had the poorer outcome. For the same corals, had much lower fertilization success when it was windy.